want to take the time to explain a little bit more about tonicity and the impact of tonicity in our physiology. Obviously, you have the representation of the three types of tonicity in these three beakers, but keep in mind that our body is not a beaker. However, these three scenarios can happen in our body as well. Now, tonicity is basically defined by the concentration of solute in a solution. But based on that concentration of solute, we can have three types of tonicity. These three types of tonicity are isotonic, hypertonic, or hypotonic. Let's start for the isotonic. Why we define that this solution where the red blood cell is immersed is isotonic, means that the tonicity or the osmolarity inside the cell is equal than outside the cell. You notice that we have 25% of sucrose inside, 25% of sucrose outside the cell. So based on the concept about the water, because it's extremely important to keep in mind and repeat it, and also you will be exposed to this material on and on and on and on through the following chapters. What happened? Which one is the behavior of water? And that one is the key in our physiology, because water always will move by osmosis, but is driven by the concentration of solute in both areas of the cell membrane. If we go back to the type of movement that is osmosis, osmosis is a type of diffusion. It's simple diffusion, but it's the diffusion of water, not the diffusion of other molecules that can diffuse through the cell membrane, like oxygen, carbon dioxide, or liquids, some liquids that are not water. But certainly, in general topics, the molecules that can cross by diffusion without using simple diffusion, without using one coin of energy or ATP, are gases and nonpolar molecules. That one is the basic. Water has a different characteristics, because in the case of water, osmosis is defined as the diffusion of water. It's not the diffusion of other molecules, it's the diffusion of only water. And from the PowerPoint, you study that water moves through the cell membrane freely, even when it's lipophobic, because it's using that unique protein in the cell membrane that are called the aquaporins. These aquaporins is a protein that is customized and is really very tiny little mini, only used by water. Now, what happened in our physiology is, excepting for one scenario, water always will move towards the higher concentration of solutes using the aquaporins. Blind is like a blind date. Is over there we have a higher concentration of solute, water will move blind over there. If it is good or bad for our physiology is another conversation. But in many cases, water will move towards the higher concentration of solute and is damaging our physiology. Now, in isotonic solution, this one is the solution that is iso because contain the same concentration than inside the cell. Now, let's talk about the scenario when it's a hypertonic solution. In a hypertonic solution, that it can happen in our body. It's not just in a beaker. In normal concentration, this one will really happen in our plasma pretty much all the time. But it's the ideal scenario that our body will use all the tools or all the mechanism to keep it in this way. And we analyze how we keep that scenario, because that solution that is in this beaker is represented by our plasma. Ideally, our plasma is isotonic and is the ideal of the osmolarity of the plasma, osmolarity of the extracellular space, and the osmolarity in the cell. What is that magic number? Is positive 300 milliosmoles. And around is average 300 milliosmoles, but they are positive. Now, what happens in case that plasma become hypertonic? Is hypertonic the solution? The solution in this case is plasma. Now, the same erythrocyte is immersed, or our plasma has changed. How can change the plasma? For example, very simple scenario is, and it's part of the exercise that you have in the PowerPoint, that you have a graphic to play with, is in case we have a meal with a really high concentration of sodium, right? Or salt. Sodium is salt. That in general is all the fast food or the processed food that we can buy in the supermarket. You can buy, I don't buy. But certainly that kind of food is high in sodium. So I invite you to remember, when you go to that place and have a meal from Taco Bell or Burger King or any fast food that you choose, obviously you get satisfied for a long period of time, long hours, basically because it's a high concentration of lipid molecules, but also immediately you feel thirsty, and instead to ask for water, you ask for a soda, right? So it's even worse, so you mess up completely. But certainly keep in mind that the first sign that your body gives you that your plasma has changed osmolarity is thirstiness. You are pushed to drink something because you feel thirsty. Yes, okay, the ideal drink that you are going to take is water, but you can smooth the thirstiness with other drink. Ideally is water. With water, we balance this out. Now, let's keep in that scenario. When you have this plasma that increase tremendously, what happens if there is no homeostasis mechanism that try to balance this out? That cells immersed in this scenario, in this case it's erythrocyte, in plasma, could be the erythrocyte, could be a platelet, could be a white blood cell, the 3 cells in plasma, but all of them behave in the same way. All of them, they have certain amount of osmolarity as well, but if the osmolarity in plasma increase above the osmolarity inside the cell in this environment, in this solution that is a plasma, we'll try to donate water, because water will move blind towards the areas of higher concentration of solute. It doesn't matter if it is inside or outside the cell. Water will move blind. It's a day blind that doesn't think too much, and it's really very full. Sometimes it's so silly that affect tremendously our physiology, but is how it behaves. Now, in this scenario, the cell will donate water because water moves towards the areas of higher concentration of solutes. And remember, water is a simple diffusion of water. Water is a simple diffusion, just that, like any other diffusion molecule, but this one is specifically for water, and water uses the famous aquaporin. Now, how long is going to donate water to the solution? As much as they keep the balance, 50-50. When they reach the 50-50, the movement of water stop because become isotonic. On the contrary, if that difference is so crucial and is so drastic, so big difference, this cell will donate blood, will donate water on and on and on and on, and meanwhile is leaving water from the cell over here, that cell diminish in volume. If we can weigh the cell before and after, we can wait that the cell lose weight because lose volume. And because of that, that cell is shrinking, and the word is crenation. When the cell crenate is when the cell shrink because it's losing water. Losing water is losing weight. Now, another important concept about this topic, the cell crenate. What happened in this cell? When the cell crenate, losing water, and water. In the real life, this one could be the scenario for dehydration. When we are lost in the desert or we run a long time exercise without replenishing the fluids in our body, without drinking water, there are different kind of scenarios, but certainly you are immersed in a dehydration, or you have a severe diarrhea, for example, or you are vomiting tremendously for several hours, you have a food poison. There are tons of different scenarios by which our body is at risk to be dehydrated. If it is dehydrated, that plasma is losing volume. Losing volume is another way to define that this plasma become hypertonic because it's losing water and the concentration of solutes increase because we lose water and the concentration of solutes increase. And in this case, we can talk about sucrose or sodium or any of the other solutes, but it's particularly about sodium. Now, is what really happened. Not only for the white blood cells or the red blood cells or any kind of blood cells. It's also happened for all the cells in our body. If we are exposed to dehydration for a long period of time without intaking water that can balance this out, obviously we are at risk to die. People die for dehydration. Who are the most vulnerable people dying for dehydration? The babies, the toddlers, the kids, and the elderly, the two ends of the scale. Because for different reasons, the babies can be unstable in a couple of hours, and the elderly as well. And it's a long topic that I will describe later on in further in other PowerPoints or in other chapters. But right now, keep in mind that dehydration happens and is life-threatening, especially in the little kids and in the elderly, but in between as well. What happens with the cell is losing water. Losing water is losing the capability to produce all the metabolism that is expected from that cell. And without producing that metabolism, the cell died. So if you add or if you project that is not happening in one cell, obviously our physiology is upside down. And we die by dehydration. So crenation is related to that action if the cell is exposed to hypertonic solution. Now, let's go to the other end of the scale. Oh, I forgot to mention something, because we mentioned that, and I do remember that in the PowerPoint, and I mentioned over here that the osmosis is the diffusion of water. Pretty much what it is. A diffusion of water means that water will move from higher concentration to lower concentration, like any other molecule included in the passive movement through the cell membrane, any of those. ion channels, facilitate diffusion, simple diffusion, and osmosis is not exception. Let's keep in mind that I can prove that one. So I can use the water move to diffusion is defined as the movement of water that goes always towards the higher concentration of solutes, or water will move from higher concentration to lower concentration. It's exactly the same. This scenario, in this scenario, how much water do we have? We have 75. How much water do we have over here? We have 55, 55%, 25%, right? So where we have the higher concentration of water, 75 over here, lower over here, higher concentration of water to lower concentration of water, and is fulfilling the definition. Let's go to the other sample that is hypotonic solution. Hypotonic solution is also can happen, and we have to give very close attention when the plasma become hypotonic. And I want to ask something about that when I finish, if I remember, sorry. Because I don't have a teleprompter to read or to follow. Pretty much what I mentioned is absolutely spontaneous. I don't have a transcript, I don't have anything over here, so it's just my brain, and sometimes it's not working very well. Anyway, so the hypotonic solution is when you have the erythrocyte, in this case, is immersed in a solution that contains a lower concentration of the same solute. In this case, the solution is also plasma, and we have the scenario: the erythrocyte contains 25, the solution that is plasma contains 10% of the same solute, that is sucrose. Now, again, water is not really very intelligent molecule, even when it's a very important molecule in our life, but water will move always blind towards the higher concentration of solute. So where we have the higher concentration of solute is inside the cell, and water will move from the solution inside the cell. Because of that, water will move inside the molecule, inside the erythrocyte, and that erythrocyte will increase the volume. Increasing the volume increase the weight as well. So eventually, this red blood cell can burst. The movement of water against, in any kind of situation, either in hypo or in hypertonic, water will move until the both areas divided by the cell membrane reach the equilibrium. However, if the difference between both sides is too dramatic, the increase of water over inside of the cell, obviously the cell can burst and the cell die. So there are the only scenario that I can come out with in this case is if the person is induced to take too much amount of water. So in general, yes, increasing the amount of water about the 5-6 liters per day, the patient died. I don't know, in my 64 years of life, I don't know nobody that commits suicide just drinking water, frankly. But it happened that people who have been tortured have been induced to drink water, and people die for that. But certainly we can apply over here the same concept that water move from higher concentration to lower concentration, where we have over here, we have the 90% and we have over here the 75%. So obviously the higher concentration of water is over here and is shunt or towards the areas with lower concentration of water. Now, I the time to write over here about edema, because these scenarios can happen during edema, specifically this one. Not because it can, well, in the long run can kill the person, but also keep in mind that the concentration of solute, and that one is a topic that you will face pretty soon, that there are, and probably you remember from anatomy, but there are certain particular proteins in blood that one of the main function is to keep the osmolarity of plasma. This osmolarity is the function is to keep that normal osmolarity. That protein is called albumin. Now, the amount of albumin have to be in ideal concentration in the bloodstream, and pretty much doesn't have to be too different with the albumin inside of the red blood cell. But the point is that if the concentration of albumin inside of the capillaries is too low, and it can happen in this scenario. Why can be so low? It's not related to the intake or not of water. It's related to the production of that protein in the liver. But also can be, that concentration is also affected by the filtration of those proteins if the nephrons are affected. They have a certain kind of disorder. So there are two main reasons why that concentration of albumin decrease in the bloodstream, and that scenario become hypotonic. When that plasma become hypotonic, there is a scenario by which water, instead to the capillary, instead to pick up the excess of water produced by metabolism of the cells in the extracellular environment, pick up that water from the extracellular environment and bring it into the capillary, instead of that low concentration of proteins in the capillaries are inducing the movement of water towards the higher concentration of the same solute, and in this case is the concentration of albumin in the extracellular space. With this scenario, water will move always blind and silly and very full towards the areas of higher concentration of solute. So we are losing water from the capillaries and the entire bloodstream into the extracellular fluid, and because of that they produce edema. Edema is accumulation of water in the tissues. Now, the first step of water is moving from the capillary into the extracellular fluid, but if we allow to stay that edema long period of time, that edema is going to that accumulation of water will go from the extracellular space inside the cell and eventually produce the same scenario. So people with edema not only produce the enlargement of the area where you have the edema, that is very common in the low extremities, but also is very risky in the lungs, producing pulmonary edema. So that one is the application about the movement of water in our body and in more extent. So you have to be familiar with that, but also the book is mentioning about how we keep the homeostasis, and specifically talking about homeostasis of plasma concentration. That one is my teleprompter, is my computer. But certainly in that graphic you have different kind of scenarios. And what happened when there is the scenario is talking about the dehydration, is talking about this scenario. In dehydration, as I mentioned before, there is a reduction in blood volume. Now, when is a reduction in blood volume, by which increase the concentration of solute in plasma, we need to do something to increase the blood volume. And the best step is not to take water from the cells. That's why our body, and I want to refer you that the osmolarity of any solute, glucose, sodium, potassium, chloride, magnesium, you name it, osmolarity is sensed in the hypothalamus, and where we have the osmoreceptors. In this case, when this scenario, we lose water. For example, dehydration, or when we take a meal that is very high, contain a very high amount of sodium. But certainly, this one is the scenario when we have a plasma that is hypertonic. The first step won't be take water from the cells. That one will be the last step if the other one are failing. Realistically, what our body produces is the mechanism, one of the mechanisms that I mentioned before, that is produced thirstiness, by which we push us to drink water. But the other mechanism is to trigger the hypothalamus, so the sensory neurons sense the hypertonic plasma, and is informed using obviously the interneurons, is informed the central nervous system, and the central nervous system is detecting or is sending an order to the posterior pituitary. And if you remember, hypothalamus is next by, it's absolutely above the pituitary. This one is the hypothalamus, this one is the pituitary. So it's really next by. So the posterior pituitary secretes the ADH, antidiuretic hormone. By action of this hormone, the target of this hormone are different cells, but certainly the most important target of the ADH are the epithelial cells of the collecting tube in the nephrons. And right there, by action of the ADH, water is absorbed, and is absorbed from the tube before it's eliminated in the urine, is taken from the tube and bringing into the capillaries. And in this way, we reduce the urination and we increase the blood volume. And it's the way we have in order to compensate that abnormal tonicity in plasma. So hopefully I was a little more clear and explanatory in this topic. And I want to mention that because obviously there are certain factors that increase or that affect the rate of diffusion. The rate of diffusion is how the diffusion is running, how speed, how fast, how slow. And I want to refer to this scenario where we have a container divided by a cell membrane. And this membrane separates the two areas. I'm going to keep in this area, I'm going to keep 80% of glucose and 60% of fructose. And on the other side, I'm going to keep 20% of glucose and 40% of glucose. What is important about that? If I'm asking you, which one is gonna be the diffusion? Probably the first thought, if you study, is obviously water will move from the higher concentration to the lower concentration. So we have higher glucose that is going to move over here, higher glucose is going to move over here. The key or the question is, who is going to diffuse faster, the glucose or the fructose? And one of the factors that affect the rate of diffusion is the concentration of solute, the amount, the quantity. Concentration represents pressure, but certainly is the quantity, the amount of concentration. Now, if we have a higher concentration of glucose, and also the two areas, because when we talk about diffusion of solute, we are not talking about diffusion of these solutes, all of them, is for any single one. Now, one of the factors that affect the rate of diffusion, and in this case, pay attention that I use two solutes. I don't use water. But in the long run, obviously, that two molecules, glucose and fructose, that are monosaccharides, and monosaccharides, regardless that they need a carrier or not, can cross the cell membrane, because based on the size, it's not a macromolecule, they are monosaccharides. But one of the things that affect the rate of diffusion is the concentration. Yes. Now, let's keep separate between each other. One is glucose, the other one is fructose. In glucose, we have in one side 80%, in the other one 20%. The difference is way higher. We have 80% over here, and we have 20% over here. So that rate of diffusion from 80 to 20 is going to be faster than that one, because in the fructose case, we have 60% of fructose and 40% of fructose. The difference is not too high. That's why the speed of the diffusion is going to be faster for glucose than for fructose. So hopefully I was more clear about that, and keep in mind about this terminology, because there are different kind of scenarios. Like, doesn't have to be these scenarios. If a cell contains 45% of sodium and is immersed in a solution that contains 85% of sodium, so the numbers can change. What you have to is play different scenarios. When can be hypertonic, when can be hypotonic, and how the cell is affected. I'm not talking about the solution. The cell will gain weight, will gain volume, will lose weight, will lose volume. There are different kind of scenarios that you have to play with. This one is the foundation, now it's in your side.